So my name is Mohamed Eladawy. I am a second-year PhD student at Nottingham Trent University. Today I'm going to talk about genotypic characterization of Pseudomonas aeruginosa coming from catheter-associated UTIs in an Egyptian hospital. As we know, UTIs are accounting for more than 30% of all nosocomial infections or healthcare-associated infections. And catheter-associated is causing an estimation of 90,000 extra hospitalization days per year. And World Health Organization had targeted Pseudomonas aeruginosa, which is the little sister of Acinetobacter frobe, as the highest priority to develop a new antibiotics. So, however, if we are looking in Egypt, there is a little is known about genomic diversity about catheter-associated UTIs. Although if you are searching in literature, you will find a high antimicrobial resistance rates in clinical settings. Plus, there is a high prevalence of a multi-drug resistant Pseudomonas aeruginosa. In Egypt, like 75% is similarities coming from different clinical sources other than UTIs between neighboring countries in Middle East and North African region. This lack information coming from because whole genome sequencing is expensive in low and middle income countries. So if we are looking for a new variants for antimicrobial resistance genes, we usually depend on PCR detections of characteristic gene primers. So my research question is, what is the genomic diversity of Pseudomonas aeruginosa in relation to three terms, which is resistance, virulence, and biofilm formation in casters? And the other thing is, does this information can tell us anything about the antimicrobial resistance and virulence of Pseudomonas aeruginosa in Egypt? So if you are interested in this study is available as a preprint, online preprint, so have a quick scan on this QR code. I'm looking forward to hear about your opinion in this study. So my study was done in a 3-month period from September to November 2021. And all the microbiology lab staff had collected the Pseudomonas aeruginosa isolates in a hospital called Urology and Nephrology Center in Mansoura, Egypt, where all the Pseudomonas isolates had been purified on citramate agar and clade agar. And after that, I sent all the isolates to Nottingham Trent University for phenotypic and genotypic characterization. So I have developed different methods. The first one is looking for antimicrobial resistance rates according to EUCAS guidelines. Biofilm formation using the crystal violet assay, whole genome sequencing using Illumina from microbes NG, or MinION sequencing in-house at Nottingham Trent University. So also these are my first bioinformatics skills that had been developed during this study using CheckM, CARD, VFDB, Bacta, and other stuff. So I have collected about 31 Pseudomonas aeruginosa isolates from urinary casters. The isolates were highly resistant to quinolones, especially ciprofloxacin and levofloxacin, because they are commonly prescribed as empirical therapy in clinical settings, as we are waiting for the clinicians are waiting for the culture results to come back, followed by tobramycin and cephalosporin, and about 19% were multi-drug resistance isolates, meaning that they are resistant to more than 3 antibiotics from 3 different antimicrobial classes. According to crystal violet assays, the Pseudomonas aeruginosa isolates were highly in biofilm formation, as we have a 77 were strong in biofilm, 19 were moderate, and 3.2 were weak in biofilm formation. Once we have all genome sequencing are coming back from microbes NG, we checked for them for all of they are all of good quality according to completeness and contamination values. According to completeness, all of them are higher than 99% are complete, and they are less than 0.86 contaminated. The two bioinformatics tools, Fastani and Phyloflantry, we have used them to check the average nucleotide identity percentage for all of the isolates in relation to the reference genomes of type strain of Pseudomonas aeruginosa. So the first thing we are looking at is the clonal diversity of the isolates that have been collected from one hospital in Egypt. Previously, there is no any information in the PubMLST database for Pseudomonas aeruginosa isolates in Egypt. So I am happy that there is the first 8 different sequence types that had been reported to PubMLST database, and we have 2 of them that are high-risk clones. The second map shows us the previous high-risk clones disseminated in the worldwide, and the high-risk clone definitions is like the isolates that had multiple beta-lactamases like NDM, VIM, OXA, or different other beta-lactamases. So this map on your left, this is the genotypic information according to CAR databases using perfect and strict hits, and on your right is the phenotypic according to EUCAST guidelines. So this map shows the isolates had an arsenal of different antimicrobial resistance genes, and we have some new rebirths of beta-lactamases, especially the OXA ones, the NDM1, OXA 520, OXA 847, and different oxacillin genes. The second thing we are looking at is the virulence genes according to virulence factor database. So isolates have encoded in range from like 196 to 210 virulence vectors. And the major virulence genes that have been major abundance, which is adherence genes and secretion genes, signifying that this isolate should be high in biofilm formation. One interesting thing we are looking at is the mega-like plasmids. So we have a previous study from 2 years ago that defines the megaplasmids that have been disseminated in multiple drug-resistant Pseudomonas aeruginosa. Thanks to Dr. Garris from our group at NTU, which suggests us to looking for these megaplasmids. So there is 3 different marker genes that have been that are already in the mega-like plasmids according to the primer sequences of this previous study. And we use the BLAST and search against our 31 Pseudomonas aeruginosa isolates. And we are lucky that we have one strain that have a higher than 97 similarities with the 3 different marker genes for the mega-like plasmid. So we need to confirm the presence of the megaplasmid encoding a new variant of OXA 520. So all reads have been mapped against the different 15 reference genomes according to the Cesaris study. So we are lucky that we have a one strain that shared more than 100% when we mapping Illumina reads against the reference genomes. And there is no other isolates are sharing more than 1.8% to the genome of the mega-like plasmid. So we need to compare between the whole genome sequencing data and EUCAST guidelines. As we know, the EUCAST guidelines had defined the susceptibility into 3 terms, which is resistance, intermediate, or susceptible. So previously, the I stands for intermediate, and we can merge it with resistance into one resistance category. But after 2019, the EUCAST guidelines have been changed for Pseudomonas aeruginosa and some other microorganisms. So there is no intermediate anymore, and I now stands for increased exposure, susceptible with increased exposure, meaning that there is a high possibility of therapeutic success if we have using increasing dosing regimen or increasing concentration at the site of infection. So we can merge now the susceptible isolates into 2 terms, which is the standard dosing regimen and susceptible with increasing exposure. So we need to compare between the whole genome sequencing data and disk diffusion test results according to EUCAST guidelines. We have a 31 concordance where all genome sequencing data are matched with the disk diffusion test results, and 69% are discordant results. However, these discordant cases are not equally distributed because there's a 68 were phenotypically susceptible on the disk, but they have 1 or more resistance genes in their genomes, and 0.9% were resistance on the plate, but they have no any characteristic antimicrobial resistance genes. And this may be accounted to merging of increased standard dosing regimen with susceptible with increased exposure or I need to compare between different databases like CAR, RISFinder, or other databases looking for characteristic antimicrobial resistance genes, because there's some discordances between different, if you are using different databases, or it depends on the expression levels of the beta-lactamases inside the cell, or usually there's some previous literature showing there's a difficult interpretation for Pseudomonas and Acinetobacter if you are need to compare between the whole genome sequencing data and the EUCAST results or susceptibility results. Unlike the other enterobacterials like E. coli or Klebsiella, which have shown a high promising values for concordance when you are comparing between genome sequencing and EUCAST results. So as a summary, the genotypic prediction of antimicrobial resistance determinants showed all isolates have one or more resistance genes. And we have 2 high-risk clones that have been reported to BAB-MLST. So we need continuous monitoring to track high-risk clones and new novel antimicrobial resistance determinants. Pseudomonas aeruginosa isolates that have been collected are able to form biofilm according to crystal violet assay, and they have a major abundance for adherence genes and secretion system associated genes. The one interesting finding is that we have a one mega-like plasmid in just one isolate carrying a one clinically important beta-lactamases as a new variant in Egypt. So this study is available as a preprint online. Have a quick scan if you are interested in. Finally, I would like to acknowledge the AERON group at Nottingham Trent University, especially my director of study, Professor Leslie, and Dr. Jonathan for doing min-ion sequencing for the mega-like plasmid. I would like to acknowledge my sponsor, and finally, I would like to acknowledge Microbes NG for letting me participate in this event. I'm still waiting for another study that have 130 isolates need to be sequenced. I'm working on that now. Thank you so much. Any questions?